Oh.
Not very important for us, only look quickly. You see that when there are not much yolk, the cleavage furrow divides completely, holoblastic cleavage.
Oh.
And meroblastic yolk do not let the furrow to go up to the end.
Sure.
So different. Equal if blastomers have the same size, and unequal if blastomers are small and big because of yolk. Understand?
Yeah.
No, synchronous and asynchronous when they divide in different types. So different types of cleavage and different animal. Let's leave it. If you need, I can copy you the whole lecture. Then, this is the animal kingdom. We study only data, stones, and vertebrates. We do not study all the other animals. Because human is a vertebrate. So again, once again. Here are, you see red ticks at animals which we are going to study. You see in the bottom lancelet, small, and belongs to group Cephalocordata. Cephalocordata. If we have time, we study amphibia, birds, and mammals, but we do not, so we'll go quickly through them. So what cleavage of lancelet? Other animals not for us. Let's miss next pictures. Хорошо? Division, cleavage in different, cleavage in human. Looks a little bit like lancelet, but at the end of cleavage, we have a different structure called blastocyst. Blastocyst. And cleavage in human occurs while the embryo moves through the fallopian tube, uterine tube, to the red uterus. So then, after that, implantation of the embryo happens. So this is blastome of human. Looks different from lancelet because we have trophoblast and embryoblast. Embryoblast or inner cell mass gives rise to embryo. Trophoblast gives rise to placenta. So, because we are in a medical university, we are interested in different abnormalities. Abnormalities. Are you interested in that? So abnormalities of cleavage. Is that possible?
Yeah.
You know that homozygous twins are a result of splitting of cells. The cells do not contact, and upper line is when we have two blastocysts. And then we have two embryos, and every embryo has its own placenta and amnion and everything. It may happen that it happens later, and we have one blastocyst, but two inner cell mass, and second line. Two inner cell mass. This. And in the bottom, abnormality, when this inner cell mass does not split, and we have conjoined twins. It happens from time to time.
One placenta.
One placenta. You see that we have one placenta and lots of problems. Oh, why finished?
No,
maybe it doesn't have energy. Then gastrulation. Normally there are three embryonic layers: ectoderm, mesoderm, and endoderm. They appear in different times. First in evolution appeared two, ecto and endo, and meso appears later. The same happens during development. Different methods of gastrulation, not important for us. You see that when integration, evolution, antibody, different methods, different cell movements. But all of them we have. I tried to make animation. I don't like it very much. But anyhow, we have. Then, when we have two ectoderm and endoderm, later mesoderm appears. You see that in protostomates, prota is on the left, at the left. All the mesoderm is the result of division of two cells. In our case, human and lancelet, and mesoderm appears from endoderm. Понятно? Спасибо. No, just some pictures, how it happens. Let's see that. How gastrula looks like. Blastopore, you see gastrula and blastopore. Interesting that in protostomes, blastopore is a mouth, but in human, another name is animals becomes later it is primitive primary mouth and has lips. Interesting that dorsal lip is very important. Dorsal lip later becomes notochord.
Dorsal lip.
Dorsal lip later becomes notochord. If you are tired, tell me please.
No,
because lots of information. And dorsal lip of blastochord gives rise to notochord and is a primary organizer. Is inductor of all the development.
Oh, like nucleus.
In the cell, yes. No, gastrulation in amphibia, not for us, gastrulation in birds and mammals. In mammals, gastrulation looks absolutely different from lancelet. You see, but the result is the same. This is the embryo in the uterus at stage of gastrula. We do not discard this. We do not discard this. Our gastrulation is absolutely different from lancelet, but anyhow we have blue ectoderm, red mesoderm, and yellow endoderm. And in the end, you can see that gastrula of lancelet, gastrula of a frog looks very alike, but gastrula of birds and mammals looks different, like a primitive node and primitive streak, but it doesn't matter because in every case we have ectoderm, endoderm, and mesoderm. Abnormality of gastrulations sometimes happens. It's from this book, this big book.
What is that? Oh, sacrococcyx.
Primitive streak is a structure homologous to blastopore. And so it can have dorsal lip and can cause a second complex of organs, like notochord, like a neural tube, abnormal part in the look only that it sometimes can happen with abnormality and have this big teratoma. Sacrococcygeal teratoma. Sacrococcygeal, you understand?
Sacroada.
And teratoma.
Teratoma.
It's a tumor.
Tube.
Teratus. Teratus sounds something not beautiful. Teratoma. Ну хорошо. And now neurulation. What we discussed today. Then you will see my pictures, absolutely ugly, ugly pictures, but I'll try. So, can be divided. Organogenesis is a very long period, lasts to the end of development, and it is divided into neurulation, when three structures are created: neural tube, notochord, and somites and gut, intestines. And then organogenesis is creation of specific tissue and organs we do not discuss. You study it at histology. and anatomy. It is stimulated by induction. Look at violet cells. They produce neuralizing factor, inductor. And then ectoderm form a tube after that. But the cells below forms not a cord. Can you understand that first ectoderm forms neural folds, then neural plate, and in the end, at the bottom, gray bottom, we have neural tube, not a cord under neural tube, and somites on the sides. Nobel Prize in physiology. What Hans Spearman. Yes, what he did. He took dorsal lip, remember dorsal lip of blastoporus 1 embryo, and transplanted to another.
Wow.
And this embryo developed 2 neural tubes, 2 notochords.
Wow.
In the bottom.
Just like the sacrococcus.
Yes, the teratoma, like something like that.
So
what this experiment showed that during development, dorsal blastopore lip, do you see that in the picture, and red? This is very important cells which stimulates all the development. And it was discovery of embryonic induction. During development, tissues stimulate other tissues to differentiate. In this case, notochord, dorsal lip, which later becomes notochord, stimulates ectoderm to become neural tube. If it is too difficult, you can stay and I can stop at the moment.
It's okay.
It's okay?
You can answer.
No, once again, neurulation of one select. You see that notochord stimulates ectoderm, and later somites appears. And what we have at the end? Neural tube, skin ectoderm, intestines or gut, coelom, it's a cavity, cellome. We have abdominal cavity, pleural cavity, between mesothelium and notochord. Next pictures about neurulation in birds, in frogs, let's go through them. It's in a frog. You see that very alike, very alike.
Miss, chordates stands for
Chordates, vertebrates are a group in chordates. Chordates includes not only vertebrates but lancelet and ascidia, some primitive chordates. Chordates is more than vertebrates, but most of chordates are vertebrates, 99%. In human, looks very alike. And in the end, again, you see neural tube, ectoderm, but we have some differences in human. Very early appears amniotic cavity, because we belong to group amnions. But you see that looks absolutely alike. This is why we study lancelet, because it is easy, but result is absolutely same. This is how the embryo looks from the above. You can see that neural tube is closing first in the middle of the embryo, and somites first are formed in the middle. And then anterior and posterior part of neural tube closes. So abnormality mostly happens in this end of neural tube and that end of neural tube.
Oh.
Yes, again, we discuss abnormalities. It is a chick embryo, but the same in human, absolutely the same. Then later, anterior part of neural tube form brain. Interesting part is part of ectoderm is neural crest. Neural crest is a group of cells that do not form skin or CNS. They form P and S, peripheral nerve system.
Oh.
They migrate to the periphery, and they form all the peripheral gland hair and others, and another melanocytes, pigment cells. Very interesting neural crest.
Oh.
Not only, they give rise to very interesting structure. They give rise to, for example, septum in our heart. They produce bronchial arc. So many, lots, a big list of very interesting structures. Neural crests we do not see in lung select. It appears in evolution later. And sometimes it is called a fourth embryonic layer. Because neural tube is typical for vertebrates, not for chordates, for vertebrates, for high vertebrates, and give many interesting derivatives. So you can study it later. Don't forget neural crest. It shows how they migrate. So some of them migrate ventrally and form autonomic and sensory ganglia and adrenal medulla, and some endocrine glands and melanocytes. That's my picture. Not very beautiful. So you see some eye. There are three parts. Dermatome give dermis. Myotome give muscles. Sclerotom give bones and cartilage. Intermediate mesoderm, neogonotome, another name, gives ducts of kidneys and reproductive system. And the lateral plate splits into two. Lateral plate, another name is splanchnotom. Splanchnica are viscera, our internal organs. Somatic and parietal and splanchnic or visceral layer of lateral plate. Interesting. Do you study anatomy just now? Anatomy.
Bones, joints, muscles.
Muscles and joints.
Yeah.
So later you study viscera, internal organs. And our internal organs have two layers. One is, for example, peritoneum and mesenterium. So which covers the gut? One covers the gut. Second lines the body inside. And we have cavity. In lungs we have pleura. With one layer covers the chest, and the second covers the lungs. Between them we have scoiolome. Pleural cavity is example of cell coilome. So it's interesting, it's typical of all vertebrates and chordates to have these coilomes. Too difficult, I think. So how neural tube and somites look at microscope. In freezing and then
Freezing.
I don't know how in English this technique. You see somites they are segmented, and not neural tube is not segmented. And once again, an embryo. And derivatives of endoderm are very simple. Epithelium of digestive tube, epithelium of lungs, pancreas and liver. All the structure which are close to our digestive system. So the result is that ectoderm gives epidermis of skin and nervous system and sense organs. Endoderm gives epithelium of and eating and breathing. And all other organs are mesodermal. Circulatory system, muscles, bones, dermis, conjunctive tissue, kidneys. So most of our what is between our epithelium of gut and epithelium of skin, all is mesodermal. Okay. So once again, ectoderm. if I give the election next term, I will change something. No. So this is derivatives of epsilon. Not very beautiful. Mesoderm. The derivatives of mesoderm. Dermis of skin.
What?
And endoderm, stomachal epithelium of. But some glands. It is difficult to remember all these structures, but in general, are you tired? Not yet. Derivatives of endoderm. Liver, pancreas, I didn't show pancreas. And as a conclusion that in every organ we can find different derivatives of different embryonic layers, germ layers. Epithelium is endodermal, nerves from ectoderm, blood vessels from mesoderm. Now, when we finish this part, then if we need a break, we have a break. Cellular mechanism of ontogeny. There are 5 cellular mechanisms. Cell division, cell proliferation, migration, cell adhesion, when they contact together. Migration, when they move. Then programmed cell death. And it is normal when some cells have to die. And differentiation of cells when they develop their specific features, their shape, specific proteins. Well, cell division is not difficult. Of course, the cell divides. This is why the embryo grows. Migration, neural cell crest migrate. Most of neural crest, neural nerve cells migrate via special glial lines. And you see brain cortex, where every cell is at its specific zone, specific layer. Could be abnormalities of cell migration. Number 3, cell adhesion. For example, neural tube. First is neural cage, then cells can contact and we form a normal neural tube. Cell differentiation and cell death, cell differentiation, you can see that first cell, then different genes are expressed. This is the basement of differentiation, different activity, different expression of genes. So in neuron specific gene, even neuron, and epithelial specific gene, epithelial cells, they will be different. And the last is programmed cell death. For example, in an embryo, at first we have our fingers all together, and then some membrane, and programmed cell death give rise to. So first we have this, and then we have fingers. Not only here, in every part of embryo, only examples. So then, embryonic interaction, I think it will be difficult for you. That during development, all the parts of the embryo contact. Maybe induction and direct cell contact, and Spearman, which studied this experiment. Let's leave it. But how important is what tissue is contacted with ectoderm in a chicken? When mesoderm left, mesoderm was taken from a wing, we have this type of that feeder. Then from dye we have dye feeder. Then from food we have scales and claws. So induction, contact of tissues. During development, the different experiments when an eye optic vesicle was removed and put on abdomen skin, and second eye developed an abdominal region. So how important cell induction is, yes. Later, neural hormonal control pills. This is from abnormalities of neurulation. You remember abnormalities of cleavage were conjoined twins. Abnormality of gastrulation was the teratoma. During abdomen, we have more and more abnormalities. 5 mechanisms: proliferation, migration of cells, mutation, adhesion, retention cells. So example for all of them. No, maybe caused by different examples. For example, something very big, macroglossia. Palm is very big. Or something small, microophthalmia or anophthalmia, no eyes, eyes are small, or agenesis of the lung. Lung. Cell migration. Interesting that in the stomach appears pancreas. Tissue of pancreas developed inside stomach. At the left is the gastroscopic picture, something in the stomach. And when it was surgically removed, it appeared to be a tissue of pancreas. Cell adhesion. Many neural tube defects. You see that picture, upper picture, I was turned left to right. So it is wrong. So different abnormalities, hernia, spina bifida, or cleft lip occult. Cells from growing cells do not form structure. At the left you see an embryo at about 51 days, but you can see this defect in an embryo. This is in a newborn. Outbreak of cell death, maybe absence of anal opening.
The bottom one is what?
The bottom is an embryo. Absence of anus. Skin. Or syndactyly, when fingers do not split. Just depend by syndactyly. Чего ты не задаешь? Ладно, хорошо. And cell differentiation. No, maybe non-differentiated tissue of lungs. Yeah? Non-differentiated brain, no hierarchy, no hills.
What happened to that brain?
Huh?
The brain.
Non-differentiated brain,
is
no folds and no grooves.
you cannot use.
Yes.
这名副其实没有脑怎么样？名副其实 no brain。
brain of a fetus maybe. And up below is the brain of a newborn, but underdeveloped.